closely with, she works very closely with Austin Smith, Professor Austin Smith, that came along and gave us a presentation as part of the training scheme a few months ago now. So you're going to be talking to us about the making human embryo models from stem cells.
Simply working as it normally does. Might have to ask people. People at home, can you see Ge's presentation there? Can you see the presentation, people at home?
Yes, I can, Louise.
Yes.
Yeah.
Can you see that?
Yes.
Oh, perfect, thank you.
Yeah.
Let's get my use technology. No. So this is. So. Okay. And okay, and it's a great pleasure to be here today. And as Louisa introduced myself, is a stem cell biologist, and I moved here about 2 years ago during lockdown, and it's really nice actually to have the new opportunity to talk to real people instead of online. I think that does make a lot of difference to research and to communication. And that's two word characters, my name in Chinese. These days I always like to put them there because this name doesn't carry meaning, means sing a song, send your message out to the broader audience. But sadly, when you pronounce it Ge, doesn't really mean anything. So, and yeah, as I said, I have my small lab and the main campus, and now I have few people really. The main research team, we call it synthetic embryogenesis. Hopefully by the end of my talk, you will understand what that two words means. And so Louisa said, I work very closely with Professor Austin Smith, and I actually started my research career back to with Austin as a postdoc back about 10 years ago in Cambridge. And during that time, we worked very close together, established this it's pointed on the work. This relatively new type of stem cells, we call it naive pluripotent stem cells. And the later talk, I tell you and why cells are so special, how we can make a embryo-like structure using cells. And before I really talk to the core science, I want to introduce you with a few background knowledge and terminology. So we talk about pluripotency and the stem cells. So you might be more familiar with stem cells when you're talking about skin stem cells, brain stem cells. But the stem cell we often talk about is called pluripotent stem cells. It's specifically referred to the cells in the embryo, very early embryos in the blastocyst. This is an example of a mouse. This yellow part of cells, they called epiblasts. They are the pluripotent cells because they are the cells will make the all cell types in the baby in the next generation, the fetus. The same terminology applies to human. So the epiblast in the human embryo also is a pluripotent cells will give rise to all the somatic lineages. And contrastly, the outer cells, the TE cells, and the dark green cells, the hypoblast cells, will give rise to the actual embryonic tissues like placenta, yolk sac will support baby growth in the mother body. And so the pluripotent cells in the embryo, they are really transient, only transient existed. So during the development, within a few days, they disappear and they committed to make somatic tissues. So for us, for a long time, research history in the field, it's always a main research interest how we can really capture cells, study them. So first target is we have to try to grow the cells in the culture dish and culture them. At the same time, we have to maintain the same features as they can represent in the embryo origin. So really, the first achievement, the best example still is in the mouse ES cells, pluripotent stem cells established back in 1981. They are pluripotent and they can grow almost for unlimited time in a culture dish. And the cells, of course, pluripotent, they can make differentiate to all cell types in the body. I think this really best example by this really pioneering experiment, the one you reintroduce a single mouse ES cells, pluripotent stem cells back to the blastocyst, the early embryo. The cells can reintegrate into a developmental program and contribute to the somatic tissue body. The baby mouse is the blue colored is the cells in vitro introduced to the embryo. What you can say here, the cells can only contribute to a fetus, not to the placenta extraembryo tissues, which in the yellow color. So this really demonstrates the power of the stem cells and also demonstrates the definition is pluripotent can only contribute somatic tissue, not extraembryonic placenta tissues. So and the research progress, and we also learn these days, pluripotent stem cells is not a single type at all. In the embryo developed within a day or two, the pluripotent cells exist in slightly different flavor, but they do exist in different slightly different manner. Like that in 2007, another type of pluripotent stem cells were derived. We call the prime pluripotent stem cells. Represent to a slightly later stage of development, corresponding to yellow, later stage of epiblast stage. The reason I want to mention this is because this has to be a huge controversial. So human ES cells were established back in 1998. You can say already 70 years after the mouse ES cell were established. We always ask how we can get the human cells, why we couldn't get the human cells, and then we got it back in 1998. Not the way means just scientists. But you can say the human ES cell very different from original established mouse ES cells. So now we realize actually human ES cells we commonly called used in the field, they are more similar to prime stem cells that are very different from real mouse pluripotent stem cells. So this is where my research career started back in 2005. So, you know, I kept asking myself, everyone asks the same question, actually, how we can get the real mouse ES cells. This is cells I called in my talk naive pluripotent stem cells. So I don't want to go into detail, tell you how much we suffered with the story, but in the end, we made some breakthrough in 2014. The two seminal papers described first established of naive-like stem cell, but the culture was not very stable. Cells that can only grow stable for a few passages, short time they collapse. So we, in the past 6 years, myself, actually, this is all my own work, that we spend a lot of effort to really improve the culture system, improve technology, so how we can capture cells. Luckily, until 2 years ago, we were able to establish various methods that will allow us to capture the human naive pluripotent stem cells directly from embryo, and we can also use reprogramming technology, derive cells from a skin dermal fibroblasts or somatic tissues. We can also use a technical resetting to convert a conventional human ES cells back to naive state. And the cells often be called PXGL cells because this is the formula we give to the name of formula we give to culture the cells. And so the cells are pluripotent, of course, because they can be in the culture, they can be induced to make different somatic cell type. You can see here represent the three major somatic lineages. And what really unique and novel about this human naive pluripotent stem cells, they can not only be able to make all the somatic cells, they can also make extra embryonic tissues. I mentioned in the beginning, mouse ear cell cannot make placenta cells, but human naive stem cells, they can make all the cell type in the human embryo, in the blastocyst. This is where my talk today is based on. So it's really unique stem cells. They have this potential to treat every single cell type in the baby embryo. And so this is, you see here, it's not a long time ago, about 2020, we started this project, asking how we can harness this potential to really make a 3D embryo-like structure. And my colleague Yanagida started this project together with myself, and we formulate an approach which kind of simulate how each cell types develop in the embryo. And this approach allows us to starting with about 100 cells, and we culture them in the tiny wells. And then this video show you here. You can see here, we started with cluster, and then the cell changed cell type, and then in the middle started cavitating and expanding. The final product is like watermelon-like structure with 3 major cell types, which represented blastocyst or human embryo. And they not only look like embryo, you can see here, this is by technology allow us to look at localization of different cell type in the embryo model. The study confirms this embryo model doesn't have the 3 major cell types exist in the real embryo and localized in the right place. And we also use a technology called single-cell RNA sequencing, which will not only look at just a few markers, we look at the whole gene expression profile in the single cells in our model, and we compare to the same technology, we compare to the cells in the real embryo. So basically, we collect 100 cells in your model, 100 cells in the embryo, and compare each individual of them. So this plot tells you overall the model cells and the real embryo cells, they very similar and they cluster to each other very well. The red is model from model, blues from embryo. And also the bottom, this analysis is really reassuring us this whole model does contain the 3 primary cell lineages and accurately enough to really allow us to confidently use this model to pursue further research. And now we can really make this embryo model in large scale, basically in a small, tiny wells, like similar size of your fingernail stick. We can make 100 in these tiny wells. So technologically, this really can provide standard experimental system tools, allow people to use them to do research on them. And we also have some results to suggest we can culture the embryo in a culture dish for a few more days longer to allow us in the future study later stage of embryo development. So this just highlight a few features which characterize the later stage of human embryo development. And so just briefly summarize what I talked to you. We could create this human embryo model, which not only morphologically similar to real human embryo blastocysts, they comprise exactly they comprise correct type of cells and locally right position, and they have transcription identity very similar to human embryo as well. So, and what really this model can allow us to do, which we normally cannot do with human embryo. For the first issues, you really cannot really do real research with human embryo, right? ethical issue, and that's the first reason. And then B, because you really just know enough from embryo, allow people to seriously do research systematically. It's a model and almost can provide us a limit, restricted sources, and now actually more standardized. And most importantly, because we're starting from cells and we can genetically modify cells really to design experiment to ask the questions that we wanted to ask. I just give you an example here. This red and the green color, they represent two different cell types, originally starting from one. And the cells were genetically modified to carry two fluorescent reporter to indicate the cell types in real time. So this allows us to record the cell type change movement during the real time development use microscopic technologies. And so, I mean, I was always asked what you can do with this model, right? And I have to say, this is really just early stage of research, and we only got this last year. So, I mean, what you can do with it, you imagine, what you think about you can do with a real human embryo, but you can't do it, right? This can really just use as a replacement in the research, and research you can think about. For application, maybe, for drug toxicity, previously you can only test on the cells. Now you can test in the real 3D structure. And for new drug screening. And what we'll be talking with clinic partners is you can use this as a training test tube to test new technologies you want to use in our clinic. Previously, you need some improvement or new application. You can always test this model before you move to real embryos. And for research side, and there's two key interests. The first is because previously the major early pregnancy failure because the embryo cannot implant to the uterus. However, to study the failure of uterine implantation almost impossible because it involves using mother, right, and embryo. So this model really allows us to really in the future develop a in vitro co-culture system to study the interaction between the embryo, this part, and the endometrial tissue from the uterus. So my own lab really is interesting how an early stage embryo was made, started from one single cell type. And you can see here, this is briefly just highlight a few key features during the first 7 days of embryo development. You started from a unified cell type, and during these 7 days, first the one cell type make variable type of cells, and the cell type, the different cells need to move around the coordinate and localize in the right place. This has to be very precisely regulated. And we interested to understand each individual event. We also more interested to understand how these 3 events are coordinated, precisely regulated. I think because this really powerful model system allow us to address question more systematically, use genetic approach, biochemical approach, bioengineer approach, even mathematical approach. We can use mathematical modeling to understand deeper the basic principle used to in a biological complex tissue organization. This can even be applied to other somatic tissue, 3D tissue formation. So because we don't use real human embryo, we use embryo model. So that's how I call it a synthetic embryogenesis. So in the end, it's a lab model, it's no real embryo. Okay, so I just close my talk there, and hopefully you can pick up a little bit of information from my research talk and be useful.
Are you happy to take some?
Of course, yeah. Okay, thank you.
with the full. People's proposal.
Mhm.
Transforming it into an embryo along the path. What are the ethical implications? How controlled are you? You're very heavily told to how far you're allowed. I mean, this is
Yes.
really quite amazing.
Yes, that's
I know you can do a lot of things with stem
Yes.
cells, but
that's excellent question, and I was often asked. Yeah, it's very exciting and also very scary topic. So far, the reason why you say I always this cartoon show you this end point, we are allowed to do research. After that, we are not allowed. So we're only actually not we're only allowed because if we work with real human embryo, we are only allowed to work the embryo, culture the embryo until the stage before gastrulation. This is very special terminology. Just before the somatic tissue started to specify this baby, the embryo is still embryo, no somatic tissue, no neuron at all. Basically, it's a very simple structure there. So because our research, as I know, is really new. So there's no law or something that you cannot do this, but we just cautiously, we always say we won't do any research, culture them beyond that stage, just comply to the same regulation applied to the real human embryo now. But I think, as I said, it's still very, overall they're very similar, but the windows are very different. So, in the next research, big chunk of research in the future will be focused on how different, actually, now we know very similar, how different, whether it's really essential or necessary we treat that as a real human embryo, and what is regulation, this is discussed now in a more higher authority.
this is a fairly simple way. You know, we can develop into embryos.
In theory, it could, but in reality, that won't happen. You cannot test it because we're only allowed to culture until that stage. So this is a theory cannot really never be able to test it properly. But in mouse, similar research have been done with mouse. Yeah. Yeah, theoretically, if some dodgy people decided to implant this thing to some recipient species, yeah, it can cause problem. I completely agree. That's why this do need properly regulated. And yeah, but can I comment? Maybe. I completely agree with you. That's why this kind of research is very exciting, but you have to be properly controlled and then perform in an ethical way. And but imagination of they can make a baby, I think it's a bit overvalued because similar research has been done in the mouse for a while. The mouse embryo structure, of course, there's a reason why, because the mouse structure didn't use the right type of cells to make it. The mouse you can test, you transfer in the mother, right? And then they can never develop over a couple of days. So just still artificially, very artificial. So yeah, sorry. I think
No, it's okay. the beginning, you said you've got this very naive,
Yeah.
that within 2 days, you flipped away into being something different and more complex. They were like fleetingly present.
This pluripotent cells is that
Yeah.
one? Yeah.
So,
Yes, it's correct.
right. And so I'm quite interested in your research process now, because years ago, so we are now, this fascinates me.
Yeah.
At time, was there a very long time?
So this is, long history of people work with mouse ESL. That's actually won the Nobel Prize back to. So you mean my work started with a human. I think the point you raised, I'm very glad you raised that point, because when I started my postdoc, I thought, oh, it's easy, because I studied in a lab, really, the best lab work with mouse ESLs. We got all the knowledge. The human ESL must be very similar to mouse ESLs. I also can finish my postdoc in 3 years and move on. It took me 6, 7, 8 years. And because the knowledge we learned applied to the human ESL was wrong. Based on some assumption, I can use mouse ESL culture medium, formula to capture the human ESL was completely wrong. And yeah, the first really 6 years, you turning around, you don't ask why it was wrong. And we even added more complicated because other people write published paper. This is why this human naive stem cell research field was a whole bug, because other people starting published paper, reported cells, human naive cell reported again, similar to mouse ESLs. And I, as a junior researcher, I knew it was wrong, but I couldn't argue because published in Nature, right? Sometimes really high impact journal. And really, took myself really years research and step by step to improve the formula. Really back to 2019, myself was quite more confident, say this is real cells. We can really use it to do something. So this is where we got now. It's suddenly become quite hot. People believe us now. We got the real cells now, and then there's value of the cells as well. yeah. I know. So, and then, yeah, once the research field was people starting to publish really mixed information, it's really hard to clear it. And I actually, I may sound like little cell, actually. And one moment, there's paper published in real high impact journals that the human naive cells is full of mutation, cancer mutation, published in cells, stem cells. And I started my career back into my independent lab 2020. I had to make them retract the paper. So the paper was based on completely wrong method. And then during lockdown, we re-analyzed data and then proved all this cancer mutation was contaminated from mouse cells, because the human naive cells was co-cultured with a layer of supporting cell from mouse. So this 20 cancer mutation published previously in the high impact journal, it's all just a mouse gene. Yeah. So how people without even, I just.
Well, that's the industry.
Yeah. I'm sorry. Maybe should not mention that pain. I'm just thinking it sounds like very easy, but it's a long journey for us to really starting from nowhere to come here. Yeah. But I still would like to say, when you're talking about this, how amazing, how dangerous, as I said, now we know this is very good, but really one big question is how good. There must be some problem. There will not be exactly like embryo. Now we probably have some time really to investigate more what's the real difference. To what extent you can use it for research, even to what question you can use to address properly. what are the limitations that has to be understood properly before you really think even more fancy applications.
While that journey to understand.
Pardon, I think, can you please repeat your question first?
Say that this feels like you split the atom.
Mm. Mm.
Atoms. So while you're developing the journey,
Mm.
is the research
I'm
My understanding of these things, you probably submit them to the journals
Mm.
and it goes for peer review and it gets published. So I think what we then rely on is government regulations and ethical conditions on what
yeah.
we can or can't do, is my understanding.
Yeah, no, obviously, because even we work with stem cells, we have to apply a license to work with the stem cells. So we have to state very clearly what do we do. And but this type of in vitro, is kind of generally called organelle formation. And until I tell you this is what I achieved, before that, you really it's just tissue organoid. And of course, when you get to the stage, then it has to be put on the table. What is the limit? As I said, what the limitation, what you can do, what you cannot do, what even more dangerous, because you can say the whole system quite simple now, because once you got real cells, the cells will do the job, and as long as you have properly controlled your experiment, you can make this happen. What even worse, because I didn't tell you this, because this is our work, and in the parallel, there's a couple other people published similar saying, right? They claim they make the same thing, but if you look at the analysis now, that they have similar structure, but they don't have a similar cell comprise the same similar cell types as with the embryo. That even causes more controversial future, even for research, right? Everyone can claim they can make this, but the quality, what's inside of your embryo, people just, public eyes got drawn by the beautiful pictures, the 3D spheral structure. They don't know about the details. I think that's even more dangerous. Yeah. Sorry.
We're running out of time. Thank you so much. Just while Louisa is swapping, I just the people online, if you want to ask any